Okay, well, thanks for joining me for this webinar. I am going to talk about large particle flow cytometry, which is sorting and imaging of cell clusters and small model organisms. My name is Deborah Frankel. I work for Union Biometrica, and Union Biometrica has designed this technology for large particle flow cytometry. And so just in short, I'll go over what I'm going to talk about today in this webinar. And I'm going to focus on the latest model of our COPAS VISION as my example for our technology. I'm going to go into the principles and relevance of large particle flow cytometry, or LP-FACS. I'll talk about features of our COPAS VISION and the FlowPilot software that is operated with the instrument and also for analysis. And I will mention our automation option with our automated LP sampler, which is an accessory that can be purchased with an instrument. And then the fun part, where I will talk about interesting things that people are doing in the field with our technology. And then I'll be able to answer any questions that you may have. So getting started, the principles of our large particle analysis and sorting are based on sorting multicellular organisms such as nematodes, insects, and fish larvae, small fish larvae. Cell clusters, colloids, and organoids can also be sorted, and some people are also analyzing and sorting seeds such as Arabidopsis seeds, bead, and pollen. And there are many more sample types that we could talk about that are either too large or too fragile, such as adipocytes and hepatocytes for traditional flow cytometers. And those are samples that can be analyzed and dispensed with our technology. So how is it that large particle FACS can change the way researchers like you approach their research models? And I think this can be really a game-changing technology for a lot of researchers in that it allows for collection of data for every object in an experiment automatically. It allows for rapid analysis of large quantities of sample and you can set up screening experiments in a way that's efficient and more time efficient than manual manipulation. So the way LP-FACS works is much like traditional flow cytometry. Objects are focused through a quartz cuvette or flow cell using a sheath buffer, and one object at a time will pass through the beams of a laser and be detected, and signals will be detected. So our vision instrument also takes bright field images of objects as they pass through, and that's unique to this instrument. And as far as sorting is concerned, this is where we have a uniquely gentle way of dispensing sample objects that allows for collection of fragile samples. So, and the way this works is sorting is done as sample is flowing through the flow cell, an airstream is constantly diverting the sample into waste or sample recovery. And when an object of interest is detected, that airflow is briefly paused just long enough to allow a drop containing the object of interest to fall into a collection container, be it a bulk container or a multi-well plate. And this allows for efficient enrichment of rare populations in a sample, sorting out a pure sample out of a heterogeneous sample, and also allowing for pure one cell per, or one object per well in multi-well plates. And so features of our COPAS vision, our system is optimized for automated analysis, dispensing, and bright field imaging, as I said, of multicellular organisms, of samples that are too fragile for traditional flow cytometry, as I said before. And analysis and sorting can be done of objects that are up to 750 microns in diameter. And this is with our COPAS vision. Larger samples can be run in some of our other instruments. The vision can be set up, configured for 4 excitation lasers, and detectors are used to analyze scatter, size, optical density, and fluorescence. Acquisition and sorting is done using our software, which is called FlowPilot, and this comes with the instrument, and FlowPilot can, I'll get into it, but can also save files so they are compatible with other common flow cytometry analysis software. And the parameters, again, we can sort by size, optical density, scatter, and fluorescence with these instruments. So getting into what our FlowPilot software looks like, it is user-friendly software. It looks much like some other softwares that people who use flow cytometry might be familiar with. It's used during live acquisition of sample. You can see that data can be displayed by histogram, by dot plot, where events are represented by dots on a graph based upon the intensity of the parameter on each axis. And here you see an example of a bright field image taken with the instrument. And beyond live analysis, our software is capable of offline analysis, which includes plate views and profiler views. I'll get into profiler view in a second. And plate view will show you where each, what event was dispensed into each well, and you can see the parameters from each event based on the well which it was sorted into. And with the automated sampler, you can also see what came out of particular wells from origin. And image processing can be used based on the region of interest, which would be your event in your sample, and image processing data can be used for further analysis of your samples and populations. So getting into what profiling is, I mentioned profiling. This is something that's pretty unique to our software. Our profiler gives a visual representation of a 2D scan of objects as they pass through the laser. So if you look over here where the laser pointer is, you can see the different lines here on the histogram. The blue line represents the optical density of the sample. In this case, it's a nematode, a C.elegans, and over the length of the organism. And here we have red and green fluorescent channels represented, as you can then see where the fluorescence is expressed along the sample. And with this, sorting can be done based upon the peak heights of fluorescence. Also peak width of fluorescence can be used, as well as number of peaks. So one can choose to select only objects with 2 peaks of at least a specific height, or peaks that only objects that have a peak in a specific location along the object. So in this instance, perhaps the ends of the object, in another instance, perhaps the middle. And then offline analysis, retroactively, peak area calculations and peak ratios can be calculated and used with this software. So it's a really, really encompassing software for doing data analysis. I mean, here's an example, a fun example of profiler at work. We have here a fluorescent image of C.elegans, which express GFP in their seam cells. And you can see in the profiler, you can see a peak of fluorescence, which would correlate to each of those spots on the worm that had passed through the laser and detectors at this point in time. So you can see a peak for each of those spots and how intense each of those is. And that's our profiler. And lastly, before I get into what people are doing and going over some data that's out in the field, I would like to mention that we also have a large particle sampler, LP sampler, which allows users to collect sample for analysis from multi-well plates or tubes, Petri dishes, and it can wash samples from agar plates. And another really useful feature is that buffers or treatments can be added to the wells just prior to acquisition. So something like an agonist or an antagonist can be added to each well before acquiring the sample from it. So for those types of screenings. So that is an accessory that can be purchased with the sorter. So now we can get into some more of the fun bits of the talk, where we get to see what people are doing out there in the world, what we're doing with our technology. And this is just a small sample of some data that's been generated using the Union Biometrica technology. And one very exciting example is work that's being done by the Kagan lab at Mount Sinai. And this was discovered using Union Biometrica technology to help screen compounds using a Drosophila model in cancer therapeutics. And this is further developed by AstraZeneca and has been approved by the FDA for medullary thyroid carcinoma. So this is really exciting work that's being done. And what the Kagan lab has done is they are able to take a biopsy from a patient and analyze the entire exosome of that biopsy and the patient, and discovering the mutations in the tumor, they can engineer those mutations into fruit fly avatars. And using those avatars, all FDA-approved drugs can be screened on up to 500,000 different avatars. So they can screen large numbers of drugs on a patient sample. So very personalized medicine. And based upon the efficacy on of the drugs on the avatars, treatment recommendations can be made. And this is showing very promising results. And I would definitely encourage people to follow this work. Moving on, other interesting work that's being done using the Union Biometrica technology. This lab at University of Michigan, the Jacob Lab, is using C.elegans as a model for stress resistance as related to lifespan. And so what they're using our sorter for is to sort C.elegans, which express a GFP biosensor, a redox-sensitive biosensor, and they, based upon redox potential, can sort the different populations from these worms. And they can use this data to then correlate this redox potential in the worms during development to longevity. This is also really interesting work that's being done out there using our technology. And just related to that, I just wanted to show a little bit here of imaging of worms and how worms can be gated and sorted for their uses, so and other uses. So worms can be sorted out to be separated from eggs and also from any debris, etc. Worm populations can also be further sorted to the different larval stages using size and optical density parameters of with our technology, and people are doing that as well. So that's how that works. And moving on to another interesting research, there is a lab at MGH in Boston at Mass General Hospital, and they're exploring potential use of retinal organoids for treatment of retinal disorders. And so what could be done with that would be, as organoids are grown and differentiated, relevant organoids based upon markers would be sorted out for further growth and differentiation and potential use in transplantation to treat these retinal disorders. And just to show how some of that looks with sorting out organoids, you can see that with each organoid, an image is taken, and the image can be overlaid with the profile data. And again, you can see that gates can be made based upon peak height, on size and optical density of the sample. And once dispensed, it's an image is saved of each dispensed organoid within the well that it was dispensed to. So this can be used both for tracking and confirmation, as well as quality control of sample after the fact. And in addition, each of those organoids that has been dispensed, you can also see in a plate view, so here representative of the wells of the plate, the profile and the profile of each of those dispensed cells, which give organoids, which gives an idea of the fluorescence, etc. And then that can be confirmed with fluorescent microscopy, looking at that plate that has been collected. And I'd also like to point out that in sorting out spheroids and organoids, that with our software and with this technology, it's relatively straightforward to be able to sort cells or cell clusters of interest and gate out objects that are not necessarily of interest. So perhaps organoids that are, or spheroids that are clumped together, single cells and debris can all be gated out quite easily. And gate can be selected to dispense relatively uniformly shaped objects. And this can be done, again, exponentially faster than it can be done by hand without this technology. So next I'd like to talk about what I mentioned before of fragile cells or fragile samples being appropriate for use with our technology. So, for instance, cardiomyocytes, up until this point, have proven more than challenging to dispense and get live cardiomyocytes back. We have a customer, a group that has published their work working with cardiomyocytes and sorting them using our technology. And what they were able to show and what they saw was they got a high viability of rod-shaped cells back, whereas with conventional FACS, they were not getting viable rod-shaped cells back. So our instrument is really gentle enough to sort out these very fragile cells, and they found that these cells were viable for culture, and they remain functionally competent after sorting. And this is a publication from 2019. And this is a group at Johns Hopkins that is doing this work. And I'd like to also use the example of adipocyte sorting. So we've been able to sort mouse adipocytes onto, in this instance, they were sorted onto cover slips for imaging, and cells that were sorted into wells and bulk collection containers experienced a 70% or more intact and viable recovery rate, which is just not possible with conventional flow cytometry for these cells. Yeah, so given all of those examples, I'd just like to go through a summary of everything that I talked about. Just a reminder that our COPAS VISION has 3 different flow cell sizes, 250 microns, 500 microns, and 1,000 microns available, depending upon the needs of any given lab that would be purchasing. Our other instruments also have the option for a 2,000 micron flow cell, if that is something that is absolutely necessary for a lab. The Vision is configured for up to 8 PMT channels, up to 4 lasers. Bright field images are taken of the objects, and analysis can be used for offline analysis, image analysis parameters, that is. Real-time profiling of objects for analysis and sorting is a key feature, and sorting and dispensing based upon size, optical density, scatter, and fluorescence, as with traditional flow cytometry, is what is done with our technology. And you can collect sample into multiple different types of receptacles. So that's the summary of the main features of our organism. I hope that this was an informative talk, and I would love to field any questions. Thank you.